Welcome everybody to our Vaccinium CAP webinar series. My name is Amaya Tucha and I will be today's webinar moderator. Today we will be discussing of buts and beats, a QTL meta-study identified stable QTLs for berry quality and yield traits in cranberry mapping populations with Andrew Maule. Andrew is a PhD student in Dr. Juan Salapa's Cranberry Breeding and Genetics Lab in the Department of Plant and Agroecosystem Sciences at UW-Madison. We are really excited to have him with us today, so please join me in welcoming Andrew. Just a little bit of some housekeeping. Just please keep your audio and your video off. If you have any questions during the webinar, please feel free to just post them in the chat and we will ask the questions at the end. Andrew, the floor is yours.
Okay, thanks everyone. I'm going to share my screen. Let me just make sure. Share the right thing. There we go. Okay, can everybody see this?
We can see it, but it's not in presentation mode yet.
Yeah. Okay.
There it is. Perfect.
Okay. So Maya already introduced the title of the paper. It's in summary, it's basically a very large scale like condensation of a lot of different traits that were collected in Cranberry and mapped QTL, and then looked for stable QTL across studies and years. But to start, like the workflow here is first like looked at trait correlations. So there was a suite of different traits that we looked at, and just kind of look at kind of the intertrait correlations, and I'll kind of go into why we look into that when we get to it. Then from there, in order to do the QTL fitting, we're dealing with two experimental populations, or I should say breeding populations of cranberries that were not planted in an experimental design, but we do have linkage map data on these two populations. And so we were able to use the marker data to kind of enhance the statistical models. And so we use mixed models to kind of fit and generate breeding values or BLUPs that were then subsequently used for mapping the QTL. And then in order to, let me just make sure that I have, let's see. Oh, it doesn't want to do it now, but it's fine. And then we did the QTL, we did the QTL mapping using a variety of different approaches. We did a single QTL mapping using interval mapping, and then also a model fit selection approach. And the QTL were mapped across 4 different fitted mixed models, one for each year, and then also one across years. And then from there, we identified stable or Meta-QTL. I like to call them, they're like synthetic QTL, or I have a lot of different names for them, but like the principal QTL are the ones that you traditionally map, and then the Meta-QTL are basically a synthesis of those principal QTL that represent, like I like to call it durable QTL across years, across studies, and then also across what I call trait blocks. So on to the traits. As I mentioned before, there was a very large suite of traits collected here. 21 traits in total were upright traits. So I think everyone here is kind of familiar with cranberry morphology, but I'll kind of just quickly go into the anatomy and morphology of cranberries. But there's reproductive uprights and vegetative uprights, and then there's also the runners, which basically kind of, sorry, I don't have a figure here for this, but like the runners are just, like they say, they just kind of run along the ground, and they're mostly vegetative, and they're just for asexual propagation. And then the reproductive uprights are the ones that produce flowers. And so the traits that were collected, our lab was sent 10 reproductive uprights per genotype. So there was 2 populations. I'm going to just kind of go forward here because I want to show the populations. So these are there were two populations that were cranberry breeding populations that were assessed. They were located or are located, I should say, in Chatsworth, New Jersey. And they were sampled over 3 years. And what happened was some of the staff in New Jersey sent us basically samples of uprights. So they include these reproductive uprights. So they were full uprights with berries. They were all reproductive uprights. So they were all bearing on some level. And they sent like like 10 of these uprights per genotype. So over 169 genotypes for CNJ02 and 72 for CNJ04. But in total, there were like 21 traits that were collected for the uprights. So they were broken down into different yield components. We looked at berry size, berry weight, and then a suite of different shape parameters. There were a lot of other interesting traits collected too, a little bit more subjective, but like traits like calyx, shape and calyx parameters, and then also shape categories. There was also 8 plot traits that were collected for the same genotypes, and this was collected a little bit differently. It was collected on a per unit area basis. And those included yield components, chemistry, and other quality traits. So by quality, I'm referring to things like acid, Brix, it's titratable acidity, TACI. But yeah. And then there was also 11 derived traits that kind of were derived from these primary traits. And those included just length to width ratio, which is an important characteristic for berry shape, especially in processing of sweet and dried cranberries. Biennial bearing index. I think this is true in blueberries, but and in a lot of perennial fruiting crops, but there tends to be like biennial bearing characteristics where they bear, their productivity increases, or like is every other year. So this is also true in cranberry, and there are cultivars that have been selected to reduce the biennial bearing characteristics so that they're productive every year consistently, which is important for growers. And then I won't go a lot into this because it's probably a little unnecessary for this talk, but like one of the derived traits that I created was called a berry chimera trait, which was I took template images of the different shape categories that were given to basically the people who scored the shapes of the berries, and then I created kind of like what I called a berry chimera, and then from that image, like that berry chimera image, I collected a bunch of different shape traits that were more numeric instead of just qualitative. Okay, so I talked already about the populations, 2 populations, New Jersey, 3 years of data per population, and 10 uprights per genotype. In addition to, like I said, there were 8 plot level traits. This just shows, like, again, the reproductive uprights versus the cranberry plots. Cranberry plot traits were collected on a per area basis. So for the 2 populations, this is a lot of information here, but the way I want to summarize this is we looked at trait correlations predominantly to look at for downstream. When I was doing the Meta-QTL synthesis, I wanted to create these trait blocks based on highly correlated groups of traits that allowed me to kind of categorize all these large suite of traits into roughly like 4 or 5 different categories that are important to breeders and growers. So for this case, like what you want to look at here is we have I don't know. Can you see the arrow? Like if I do this, does it show up?
Yes, yes, we
Okay,
can see
okay. It
your
was
cursor.
showing up earlier, but it's not showing up quite for me right now, and I'm just kind of like making sure. So there's like each of these squares just represents kind of like a different trait block, and like some of them, some of the traits that are highly correlated are kind of, you know, trivial or expected, but um, for instance, like this trait block here represents a group of traits that are related to berry weight and yield. So it's like a yield component, but it's specifically a yield component related to like, you know, individual berry weights. And this block here includes both upright traits and plot level traits. So two different ways in which traits are collected, which is kind of important because you want to see some overlap or consistency there. We have berry shape parameters. We have length versus width, eccentricity. This is berry length. So it's a combination of berry size and shape. And then the yield components that are more related to like total yield, like on a per area basis. PAC here is proanthocyanins, and that's just an interesting correlative effect with total yield here that seems to just be kind of some kind of a linkage, you know, drag event, or if you want to think of it that way, where it's not really drag, but I would say that the proanthocyanins seem to be inversely correlated with yield in this CNJ02 population. On the right here, I just ranked in, I ranked the top and the bottom highest correlated pairwise correlations using pure, um, not Spearman, but sorry, I'm just, these are the highest and lowest correlations, I should say, that pairwise correlations between the different traits. And again, some of them are obvious, like total yields, sound fruit yields. Those are trivial, should be like, they're expected to be highly correlated. But yeah, like I won't like overwhelm us with too much of this here because I just kind of wanted to focus on the fact that we're using these trait correlations as a way of showing how like we kind of coalesced and organized these traits into trait blocks. This is the CNJ02 population. Again, we have a tight block here of correlated traits related to shape and shape and size, berry shape and size. Some yield components. UNB is like upright number of berries. Not surprising, it's highly correlated with upright total berry mass. And also total yield, which is again a plot trait. So we have upright traits versus plot traits all kind of grouped together, which is good because it's nice to see that consistency between two different kind of collection methods. From there, after doing some initial trait assessment, we fit mixed models to calculate, the genetic heritability and breeding values. But to begin, in order to do both the mixed model fitting and the QTL mapping, we used composite map that was generated in 2017 by our lab that consisted of 6,073 markers across 3 different populations. So it's a kind of a condensing of 3 different populations into a composite map that provides utility for doing cross-population QTL mapping studies. This is the general workflow from gathering. On the left, we gathered the trait phenotypes. I talked about that. We fit the mixed models. These genotype markers represent the SNP calls, if you want to think of it that way, or in our case, we also had SSR markers. But in this case, we used the SNP marker calls to calculate the additive relationship matrix. If any of you are familiar with kind of the animal the animal model when it comes to fitting mixed models. This is kind of like akin to that, except we don't use a kinship matrix from like a pedigree, but we use marker data in order to develop a variance covariance matrix that kind of builds the relationships between the different genotypes. And again, this is important for the fact that we don't have an experimental design, so it allows us to kind of, it allows us to deal with building additional, variance and covariance structures into our model fitting that kind of allows for it to fit in a better way and allows us to calculate breeding values. And then from there, like this is just kind of one representative mixed model. I also have another one that includes the across year data. The output of the mixed models, we get variant, like an estimate of the additive genomic heritability. That so that gives us an idea of like the genetic signature, the strength of the genetic signature associated with each trait. And then we also pull out breeding values or BLUPs. These are then fed as input into the QTL mapping process. From there, we gather or discover a large amount of principal QTL. And then from there, I sent them through a Meta-QTL kind of pipeline in order to condense and find the most notable, consistent, stable, durable synthetic QTL is the way I think of it. So this just kind of shows a plot of the heritabilities of a variety of the different traits. So not all of our traits had a genetic signature, so the mixed models didn't always, they didn't always find a solution. So in that case, we just assumed that there's not really a genetic signature for those traits, which is not surprising because these are two breeding populations. We can't guarantee that there's segregation in all the traits that are collected. So that being said, these are some of the most important traits here in terms of heritability, like strongest genetic signature. There's an incredibly high heritability with respect to length versus width in both populations. We have, let me see, CNJ02 here and 4. We have ULVW, which is upright length versus width. Length, which seems to be driven a lot by the length in both populations. TASI is also very highly correlated. We'll talk about that. Mean fruit mass is highly correlated, and yeah, those are some of the most important ones. So we have length versus width, so which is like I said, a shape parameter. It's important for sweet and dried cranberries. You want rounder cranberries for the processing of that. And yeah, this so there's like two different goals here in the end with these QTL. Like you can do like a kind of a marker assisted selection with the QTL you find, but also you can also use the breeding values if you want on their own, which might have some value to people making kind of these decisions, these selection decisions. So the breeding values or the BLUPs that were pulled out of the mixed models, I kind of categorize them into different traits, and I ranked them and stacked them in a way that allowed kind of to show like and allowed like breeders to kind of choose like ideal genotypes that kind of can stack multiple different traits in a favorable way. But I won't go into too much detail on that. I just kind of wanted to kind of talk about the two different ways. Most of this, though, this talk is focused on the QTL and the Meta-QTL specifically. Okay, so there were, let's see, there were three different like ways in which I calculated Meta-QTL. They were a cross model, let's see, I'm just gonna kind of skip some slides here, but like across models. So like when I say cross model, like I said, I fit 4 mixed models, 1 per year, and then one that encompassed all years. And so I had 4 models total. And I'm looking for stability and consistency in, across those different models or across the years, if you want to think of it that way. I also did, let's see, let's get over here. Cross. So I did a multi-trait Meta-QTL analysis, and those encompass using those trait blocks that I was talking about, where I use highly correlative traits to kind of build these trait blocks. And then from there, I look for groupings of them that kind of overlap on linkage groups. And then from there, I also did kind of a cross-study Meta-QTL analysis. And in this case, I used different studies from our lab, different previous QTL studies to look for, consistencies in other traits that have been looked at in the past and see how they overlap with the QTL I found here. Again, to kind of strengthen our like confidence in using some of these QTL as markers for like marker assisted selection or different types of marker panels. Okay, so we're going to go back to talking about a cross-model QTL. So when we looked at the number of QTL that were found across the different years, or I like to just say models too, because like I said, one of the models is an all-year model. What I was looking at was for QTL that were found in 3 of the 4 models. And so in this case, we had found 13 total Meta-QTL across models for this. In this case, this is populations CNJ02 is an A and CNJ04 is B. For CNJ02, there was 9 total Meta-QTL found for across the models. And I also wanted to add too that these QTL, there was some filtering criteria to kind of constrain them to look at only the strongest QTL. So I looked for QTL that had an R squared or a percent variance explained greater than 10%. So all other like principal QTL that I found through the QTL mapping, if they were below 10%, they weren't even considered as a part of this process. So for the CNJ02, we found like I said, a 9 total Meta-QTL. We found 2 for shape, 1 on linkage group. We found 1 on linkage group 3 for length versus width, and then also 1 on linkage group 9, again, length versus width. We found 1 for size on linkage group 10 for berry length. And this is upright berry length. Found 3 for fruit weight on linkage group 11. And 1 for yield on linkage group 12. SFY is sound fruit yield, so it also incorporates rot. 1 for TASI. And, I think I forgot to mention too that there's 1 right here, PFR, which is percent fruit rot. And again, these are QTL that are in the CNJ2 population that are in at least 3 of the 4 models. There's overlap there, so that you have more confidence that these QTL are actual meaningful QTL. And again, for the across model, we're looking at CNJ4. There was 4 total QTL found there. And I wanted to remark too, since CNJ4 is a smaller population, I mentioned earlier that it had a size of 72. There's a little less power to find significant QTL. So the findings will tend to be more limited. it's not just the fact that there might not be segregation for these traits in this population. It's just also we just have a smaller population size. So one for shape in linkage group 11, one for size in linkage group 11, one for fruit weight. UTBM is it's total berry mass, but a lot of the total berry mass was driven by the largest berry mass. So we kind of consider that fruit weight. And then PAC, which is proanthocyanins, which is an important, I don't think I need to explain too much about this, but proanthocyanins is an important like quality trait. It's a secondary metabolite that's often important to human health. And then, so the next, after doing the across model QTL, I mentioned that then the next step was I was looking at multi-trait or trait block Meta-QTL. In total, we found 11 of these multi-trait QTL, 7 in population CNJ02. For CNJ02, we found 2 for shape, one on linkage group 2, one on linkage group 11. These shape parameters include, again, length versus width. We have berry length, which in this population tends to seem to be what drives the length versus width. The eccentricity and another length versus width parameter. For fruit weight and size, we found three Meta-QTL on linkage groups 10 and 11. For yield, we found 1 in linkage group 12. TASI on linkage group 3. Then for CNJ population CNJ04, we found 1 Meta-QTL for berry length and length versus width, so shape and size berry shape and size parameters. And then also like I kind of combined both populations, so I added an additional filtering or constraint parameter around whether I needed to have QTL that showed up in both populations. So I'm just looking for both populations like stable QTL. The reason why I did this on some level, you might say like these are 2 different populations. Why would you necessarily look for QTL that are stable across? Because sometimes when you're dealing with multiple or separate populations, they just have different genetics. So the QTL are specific to a population. But these 2 populations do have some shared pedigree. So it's just some interest in seeing where there's some overlap there. So there's 3 Meta-QTL found for multi-trait blocks. 1 for shape and size on linkage group 11. 1 for yield on 12 again. And then 1 for TASI on linkage group 3. And then last, I looked at several different studies and I looked for consistent QTL that I could call across the different lab studies that we've done in the past on QTL. There were a total of 29 cross-study Meta-QTL found, consisting of 63 independent traits. So for the first study that I considered, it was a study done back in 2018 by Luis Diaz Garcia in our lab, and he used a software package called Gina to use like photos of cranberries to pull out color traits, and he also collected TASI data on that. Since we looked at TASI2 and as one of the plot traits for this study, I was looking for overlaps in color and TASI. So total anthocyanins, they're associated with or highly associated in cranberry with, the berry color, specifically berry red, like the red anthocyanin colors. And so in this case, we found two independent, but two independent multi-trait Meta-QTL on linkage group 3 for TASI. The second study that I looked at was another study from Luis Diaz-Garcia in 2018 also that was also used GINA like to look at look at pictures of berries and to pull out traits from those images. But these traits were more focused on shape and size parameters. In this case, we found 8 total Meta-QTL between our study and this study. 4 for shape, so linkage group 11 and 12. So 2 on linkage group 11 and 2 on 12. 4 for berry size and fruit weight. So 1 on linkage group 1, 1 on 2, 1 on 9, 1 on 11. The third study that I looked at was one done back in 2015. So it was a QTL study done by Brandon Schlautman in our lab. He was looking mostly at yield traits. Also, he calculated a biennial bearing index. And since we were able to kind of also calculate like a biennial bearing index to kind of associate how much variability there is in, consecutive years production of, cranberry, like a particular genotype, we're looking to see that we have consistent, like lower variability in yields between 2 years, any 2 years. So in this case, we found 7 total Meta-QTL, 5 for berry size and fruit weight. So 1 on linkage group 6, 1 on 10 and 2 on 10 and 2 on 11. And then also we found 2 for yield. I kind of classified biennial bearing index on total yield as is a yield trait. It's just a specific kind of class of yield. And then we have 2 on linkage group 11. And then there's also sound fruit yields, total yield, and pack. I mentioned before, like pack is obviously not a yield trait, but it kind of gets lumped in there because it's a highly correlated trait that I showed earlier with the heat map that I had. And it just seems to be like some kind of a linkage or pleiotropy related association. And then last, I kind of condensed two of the studies. And like, so before I just kind of did a pairwise study comparison from ours to some previous ones, but this one includes two of the studies that were looked at previously plus ours, and just to look and see like what kind of QTL we pull out. So we had 12 total Meta-QTL found, 4 for shape, 2 of them on linkage group 11, 2 on 12. So again, that's pretty consistent there with what we've been seeing, 11 and 12 for shape. Berry size, we found 7 Meta-QTL. So 1 on linkage group 1, 1 on linkage group 2, 1 on linkage group 9, 10, 1 on 10, and 3 on 11. This was one for berry yield. This one's for yield. I have that marked wrong, but yeah, this is a yield trait here found on the linkage group 11 across the 3 studies. And let's see, did I get shape? Yes, I got shape. So in summary, yeah, sorry, this is a lot of data to go through, but we found a little over 1,500 single QTL, 470 which were major. So major, in order to classify something a major, again, we looked at percent variance explained greater than 10%. And so these are the principal QTL. But what we wanted to do is, again, synthesize or condense into the most meaningful QTL, the ones that looked the most durable or stable across traits, across years, across studies. And so in that case, I've already kind of went through these with the other figures, but we found 13 QTL that were stable within the study across years. So these are all major QTL, Meta-QTL. Looking at the multi-trait blocks, so kind of trait groupings of highly correlated traits. We found I have 8 Meta-QTL here. There was some additional that were found sorry, I keep doing this. There were some additional that were found, but there were eight major QTL found in the multi-trait study. And then also the Meta-QTL study that looked across studies, we found 29 total Meta-QTL, 63 of which encompassed 63 single independent traits, but 29 trait blocks. This is just a snippet from one of Luis D. Garcia's papers. It was one of his genome papers in 2021, where he did kind of a candidate gene assessment of the total anthocyanins on chromosome 3. So I mentioned over and over again, we kept seeing the TACI signature on linkage group 3. He kind of did a subsequent study where he regenerated the linkage map. So it's a slightly different linkage map. So the centimorgan values don't quite overlay with the linkage map that I ended up using for my studies and the previous studies. But there is this strong evidence of this meaningful, QTL in linkage group 3 that controls berry color, total anthocyanins. And here Luis talked about some of the potential transcription factors that were in that region, that QTL region, that could play into that. I wanted to thank all these different organizations for the funding. From our own lab, Juan's been amazing mentor. He's been very supportive of me in my very long career here. Eric has helped a lot with some of the data collection and a lot of this. This all preceded me in terms of the data collection, and he was responsible for kind of fostering and helping with that. Want to thank Cranberry Creek, Cranberries. So these different growers that support our research, Valley Corp, Saddle Mound, Rutgers, so Nikolai Vorsa, he's instrumental in, of course, providing us with these breeding populations. He gave us the germplasm that allowed us to do this research and literally the materials too. The Wisconsin cranberry growers, Tom Lochner in the past, and Ocean Spray, USDA ARS, and Amaya from UW. And I guess I'll leave it at that. I have some stuff in the appendices just in case people had questions, but I'll leave it open to questions now.
Great. Thank you for that presentation. That was great. I don't see any questions in the chat, but we're a small group. So if people have questions for Andrew, I think it's just fine if you just want to go ahead and unmute yourself and ask it. If you're a little shy and you want to just type it on the chat, I'll be happy to read it too. So I'll just let people go ahead and ask questions.
Hey, Andrew, that was a lot of really good information, and thank you for sharing all of that. I had a question about the QTL for which you found for composite traits versus traits per se. I noticed in your figures that a lot of the times the QTLs were for the composite trait, but a QTL wasn't found for the the trait per se. Do you have any idea about why that would be, and can you like make any recommendations for folks who are, looking at that trait in the future, and which method you would use?
Okay. Yeah. Could you point me out where you are specifically thinking? I mean, I guess maybe it was multiple. Um,
Yeah, yeah, keep going. Well, okay, any of those are fine.
so
So the ones,
your question is I found like kind of you're saying I found a QTL here, but that were like composite traits, but they weren't found independently. Is that what I heard you say?
well, if I'm understanding it, okay, so is U, when there's a U in front of the abbreviation, is that a composite trait, or is that like when you have BL versus UBL, what's the difference? Like one's berry
So,
length? They're both
yeah.
berry length, right?
Yeah.
Okay.
So yeah, thanks for asking this. Yeah, this is just so much material. And um let's see if I can go. I'm gonna go to a zoom in one. Let's see if I can find an example that's zoomed in. It's a little bit easier to see for people. Let's see what this was I think what you were specifically referring to was this cross study here. I'm gonna go forward. So in this case, this is kind of discussed in the paper, but BL is berry length, and that was a trait that was pulled out by Luis Diaz Garcia using GINA, so using the image analysis software. So this was a QTL trait, or a trait I should say, that he found using, the digital image processing software. UBL is like an upright trait that was measured manually by us. So that was one of our traits that we measured on an upright. And I didn't really quite clarify this because it's a lot of detail, but like the berry trait, there was like upright traits, and then there was like within that category of upright traits, there was also a berry, like largest berry trait. So you took the largest berry, and that's what some of these characteristics came from. Like so, UBL is the largest upright berry length. And so it may not be quite representative of all berries on an upright, but so that might be sometimes why you don't quite see that overlap, but we still see the overlap. So it seems like, the largest berry seems, there's not a lot of berries per upright per se. So the largest berry was often the one that kind of drove that trait, but they're not always consistent. I don't know if that, let me know if that answers your question a bit, but
Yeah, that answers it. Thank you.
okay.
Well, if I may add to that, is that a lot of these traits are, again, some of them are like full plot traits that were taken with an imaging, and the other ones are like hand measured on individual berries. So one of the reasons why we did this is because in the old times, the breeders used to do the upright traits. So originally there was no plot measurements. Everything was done on individual berries. And you just had a hard time doing how many berries. So, usually people took an upright and assuming that all the objective is to have berries that are the same size in an upright. And so you would measure, the largest berry or representative berry from that upright, and you would measure 10 by hand. And so, this is in a way the fact that some of these old methods uh, are coalescing QTLs for modern methods really shows that there's power in the old methodologies, but there is probably even more power in the new methodologies. So when we're looking at a lot of these traits, they're all like fruit shape or fruit weight or fruit size or yield, or it's just different ways of measuring traits. One of the problems that we have in cranberries is that we don't have as many populations, we don't have as many years of data collection, but we can actually have a suite of traits. So this is another way, multi-trait. It's a different way of kind of coming to a consensus of what is real. Like you can have multiple populations, you can have a lot of years, you can have different types of analysis, but also you can have traits that are related. And when you see blocks of traits that are related to the same thing, that is very powerful to decide what is the best QTL. So this is where Andrew did a really good job trying to coalesce and do the multi-trait analysis and ultimately the Meta-QTL analysis that he did.
Thanks, Juan. Yeah, thanks for adding all of that. Andrew, in your figures where you show that there's like a block of a chromosome that has a lot of traits in it, I wonder, like, have you thought about doing like a selective sweep analysis? Um, which is, kind of looking at areas of the genome that seem to have been selected over and over during the course of cranberry domestication.
Yeah, you're kind of testing the limits of my or what I remember with selective sweeps. I remember looking at selective sweeps more for like kind of like usually you're dealing with like correct me if I'm wrong, but you're dealing with more inbred lines with less, there's more confidence in tighter intervals, and we just don't have that, I think, with these crosses because we only have basically one cross history like that we're mapping on. It's one history of recombination.
Okay. Yeah, good point.
So it's like
Thank you.
you need to kind of like get more crosses in to like break down and like really fine point or like pinpoint, I should say, that region. But yeah, I mean, that would be ideal. Yeah, I think that would be a great approach if we could like kind of break down or get that. Have you seen that done in any of the Vaccinium crops?
I haven't looked that closely though, and it just occurred to me right now looking at like these regions that where there seem to be a lot of traits that are important for commercial cranberry production. I'm like, I don't know if it's linkage or pleiotropy or I mean it could be either of those and also be like a signature of a selective sweep. But I think you're right also that this probably isn't the population to be looking to answer that question in.
Yeah, or
And
these three populations in general, but yeah,
Yep.
someone
Andrew,
else.
yeah, just a comment. Yeah, it's been done in Blueberry, but the point is right. So you want to use a diversity panel for doing those type of analysis.
Okay.
Andrew, a question. So for some QTL, seems like the TATSI that it's consistent across population. Do you know if you were able to narrow the region associated with the TATSI at the genomic level? Like, because you can use the recombination events of multiple population to look at the flanking marker. Have you looked into it?
I didn't look at the flanking markers for this study at all, but I did contribute to one of the Louise papers, and then like the 2018 one where
Right.
we looked at TASI and color, and the QTL in the Meta-QTL we found here, the cross study stable Meta-QTL in linkage group 3 was found around 56, I think, centimorgan. And yeah, I think looking at those you're right, like looking at those markers will be valuable because we could then cross compare them to the genome and the linkage map that Luis had for 2021. And
Yeah.
then you could see if there's at least consistency there, because we obviously can't compare the centimorgans with different linkage maps, but we can maybe compare like markers by using some kind of like I don't know, maybe you would have a better idea of like how to do that, but like some kind of blast search or something, like some kind
I
of
mean, you could use the
Passing
coordinate.
that we have this population is
Yeah,
for this study is that this is done in a composite map. So, we don't have a linkage map for each population. And so that's why Luis, in this 2021 study, he went back and redid the map using SNPs so that it would be the recombination of the population he was using. So then,
yeah.
that is probably the closest we can be, because in this population, we are using the same map for two populations. So
But I will say on that we do also have the parental consensus map you're right, like this QTL was found on the composite map, but I can back map that to
okay.
the
Well, so definitely
consensus,
or you could try
the
to
parental
use the
consensus maps
genome the genome
Yeah.
coordinate.
Yeah, exactly.
Yeah. I mean, we're redoing this too in a diversity panel. So, we're working on that for both color and TASI. So, we're hoping to get closer to that with the diversity panel. That's the goal. But yeah, I mean, how do we get closer? That's one of the questions that we're trying to figure out.
Yeah, and it's funny, Massimo, because for the 2023 paper, the plant physiology paper, on flavonoids, we and what was interesting is, I kind of did a bit of a candidate gene analysis there, and I forgot I did that back in 2022 with you, and so I could totally apply that here, some aspects of it.
Yes. Thank you.
Do we have any other questions for Andrew? Well, it doesn't look like anybody has any other questions. I'm going to paste again here in the chat where we're going to post the recording of this webinar. If you want to watch it again or you want to share with anybody, it's going to be on the VacCAP YouTube channel. We have a lot of the other webinars that we have had previously that are also posted there. And so since I don't have any more questions, I just want to thank Andrew for a great presentation. Thank you for sharing your research with us and everybody for attending.
Yeah, thank you.
Thank you.
Thanks for the questions.
Hey, nice job, Andrew.